Okay, perfect. Hello, ECI community. Welcome to the last talk of 2022. Today, we're excited to host Dr. Tsukasa Nabekura. Dr. Nabekura is a tenure-track assistant professor at the University of Tsukuba. He earned his PhD at the University of Tsukuba, and then, hello, ECI community. Welcome to the last talk of 2022. Today, we're excited to host Dr. Tsukasa Nabekura. Dr. Nabekura is a tenure-track assistant professor at the University of Tsukuba. He earned his PhD at the University of Tsukuba, and then, hello, ECI community. Sorry for the feedback there. As I mentioned, Dr. Nabekura earned his PhD at the University of Tsukuba. He then went on to do his postdoc at UCSF in the lab of Dr. Lewis Neer, studying NK cell biology. And he's been an assistant professor at the University of Tsukuba since 2014. Where he studies NK cell biology as well as innate lymphoid cells. We're excited to have him today, and thank you for being here, Dr. Nabekura. Sorry, Dr. Nabekura, you're muted, so.
Can you hear me?
Yeah, we can hear you now, thank you.
Oh yeah. Anyway, thank you for your introduction again. I'm Tsukasa Nabekura, and here is 2 a.m. right now. My brain is a bit foggy, but I will try to do my best. Today I will talk about the our recent research progress about the liver ILC1. Okay. And then I will start with brief introduction about the history of innate lymphoid cells, ILCs. Before we are born, immunologists found a very strange subset of lymphocytes which don't express T cell receptor, TCR, or B cell receptor, BCR. But somehow, when we co-cultured with this strange subset of lymphocytes with several types of tumor cell lines in vitro, these lymphocytes can show the natural site toxicity. That's why the immune cells those days called these cells as natural killer cells, named after the activity. So I'm actually the natural killer cell researcher. But when I started my research in 21st century, many research groups independently reported the tissue resident are typical NK cell subsets, actually in many tissues, such as liver, thymus, intestines, and fat tissues. And then they often reported NK-like cells in organs. Because those days, we met the many nice development of the multicolor flow cytometry or progression of the techniques of omics. That's why we could find very strange and minor populations in organs. Nowadays, almost all of them are recognized as innate lymphoid cells. And here is a consensus classification of ILCs. Right now, NK cells and the LTA, lymphoid tissue inducer cells, included the innate lymphoid cells. And as shown here, this classification mirrors TH1 to 17 classification of helper T cells. And exactly same as the helper T cell classification, the group 1 expressed TBAT, group 2 expressed GATA3, and the group 3 ILC subsets express RO gamma T as critical transcription factors. And probably many of you know, here is the driver cytokine and the effector cytokines of individual immune cell subsets. And as you know, group 1 and NK cells and ILC1 produce the interferon gamma as a critical effector cytokine, and ILC2 produced IL5 and 13. And then ILC3 express IL-17 and LTI express also IL-17 and IL-22 and lymphotoxins. And probably you can understand the function of these individual immune cell subsets due to the by these effector cytokine. Yes, so these group 1, 2, 3 ILC can elicit type 1, type 2, type 3 immune responses and then fight for intracellular pathogens, tumors in case of NK cells and ILCs. And the ILC invoke extracellular pathogen and the helminth against helminth immune responses. And the ILC3 induce the Th17 type immune responses to fight extracellular bacteria and the fungal infections. And the LTI contribute to the lymphoid tissue formation, lymphoid structure formations. And today's topic, I will focusing on ILC1. But I'm actually based on the NK cell research. And but the ILC1 and the NK cells are fundamentally very hard to distinguish due to their functional phenotypes. But because both of them nicely produced interferon gamma production, and then both of them requires IL-15 for the development. And the Tibet is also important for their development. And then driver cytokine for both subsets are IL-12. So the IL-12 is strongly induced interferon gamma production by NK cells and ILC1. But NK cells show natural cytotoxicity, especially against viral infected cells and tumor cells, but the ILC1 has little or no cytotoxicity. And then NK cells are blood circulating cells, and the ILC1 is tissue resident cells. Especially, we could see abundant population of ILC1 in the liver. And then NK cells are required EOMAS, a transcription factor, such like Tibet, but the ILC1 do not require the EOMAS for the development. And then NK cells and ILC1 express the NK receptors on their cell surface, but the NK cells express a bunch of activating and inhibitory NK cell receptors, but the ILC1 express limited kinds of NK receptors. So when I was researching in UCSF, all of us are NK cell researchers. That's why we often ignore ILC1. And then we call the NK cells a hardworking elder brother because they produce and can kill the target cells and they are circulating and travel a whole body. But the ILC1 don't move, and then they could not show any cytotoxicity in the naive situation. However, I was always wondering what ILC1s are doing in the liver. So they should have some special function, and which is unlike NK cells. And then here is the ILC1 introduction slide. They express the IL-15 receptor, IL-18 receptor, IL-12 receptor as a cytokine receptors, and they express the NKp46 and CD226 as a NK receptors. And then they reside everywhere, fundamentally, in the whole body, everywhere. And then they can elicit type 1 interferon to fight the intracellular pathogens, and then viruses, and the TB. And then they primarily produce interferon gamma after the activation, but they couldn't see any couldn't show any cytotoxicity. And then, as I mentioned already, the strong driver cytokines are 12. And here is the graphic overview of the roles of ILC1 in organs. So left side is the infection, and the right side the tissue injury. But simply said, ILC1 in organs can regulate local immune responses and inflammation by producing interferon gamma. as shown here, interferon gamma, ILC1 produced interferon gamma to fight and control the CMV infection and the intracellular pathway infections. This is good aspect of the ILC1, but in the other words, in terms of tissue injury, ILC1 can elicit inflammation, but in almost all cases through the literature, ILC1 derived interferon gamma exaggerates tissue injury in terms of IBDs and the insulin resistance, and then inflammation in these organs. And ILC1 historically were originally identified in the liver. We could see nice population of the ILC1 in the liver. However, the pathophysiological role of liver ILC1s still largely remains unknown. So in this research, the objective of our research is to elucidate the role of liver ILC1 in the acute liver injury, because in terms of acute liver injury, no one knows about the role of ILC1. And then ILC1s are innate immune cells. That's why we focus on acute liver injuries. To test the role of liver ILC1, we chose an acute liver injury model by injecting carbon tetrachloride CCl4. So because it's a traditional acute liver injury models, and this mimics the drug-induced acute liver injury in human beings. So the very first question here is, liver ILC1 are involved in the pathogenesis of the CCl4-induced acute liver injury or not? We simply injected the CCl4 into B6 wild-type mice and see the population and compare the percentage and number of ILC1 in the liver in naive mice and CCl4 injected mice. 1 day after CCl4 injection, the percentage of ILC1 decreases because NK cells increase by recruiting to the injured liver. But I couldn't see any increase of ILC1 number and percentage. However, when we analyze the activation status of ILC1, we saw the nice upregulation of CD69 from bold black line to the red line. Unlike ILC1, NK cells did not upregulate the CD69. So CD69 is the activation marker for lymphocytes. That's why these results demonstrated that ILC1 but not NK cells in the liver are activated after CCl4 injection. To consolidate the activation of ILC1, we also saw another activation markers. So we checked the CD25 expression, which is the activation marker on T cells and NK cells. And as shown here, after CCl4 injection, liver ILC1 strikingly upregulated CD25. And when we see CD25 positive population and negative population, but the NK cells don't upregulate CD25 after CCl4 injection. So NK cells don't respond to CCl4, but the ILC1 respond to the upregulated in response to the CCl4 injection in the liver. And when we compare the CD69 activation marker in CD25 positive population and CD25 negative population of ILC1s, we saw the CD25 positive ILC1s showed higher levels of CD69 expression. So these results demonstrated that liver CD25 positive ILC1s are activated ILC1 in the liver. And then we could see the activation after CCl4 injection. But more importantly, these CD25 positive ILC1 population produced nice efficiently produced interferon gamma after CCl4 injection. CD25 negative population also produced interferon gamma, but the CD25 positive population predominantly produced interferon gamma, as shown here. Here are the quantified data. Before CCL4 injection, we could not see any CD25 positive population, but after CCL4 injection, we could see CD25 positive activated ILC1 in the liver, and then in response to CCL4 injection, and CD25 positive population produced interferon gamma. So next we move to the liver injury. This time we use RAG1 deficient mice because we compare the liver injuries in viral type mice and RAG1 deficient mice, but we couldn't see any differences. That's why in this experiment, we use RAG1 deficient mice, which lacks T and B cells, and NKTs as a mite cell. Now we know CD25 positive ILC1s are activated ILC1, so we injected anti-CD25 depletion antibody to deplete CD25 positive ILC1. And after the depletion of activated liver ILC1, the systemic levels of interferon gamma decreased. After CCL4 injection, the ILC1 and probably NK cells produced interferon gamma. That's why we could see elevation of interferon gamma systemically in the plasma. But after the depletion, interferon gamma levels totally went down. So that's why these results strongly suggest the activated ILC1, the major source of interferon gamma. Very unexpectedly, when we measure the liver injury levels by plasma ALT, after the depletion of activated ILC1, the CD25 depletion antibody injected mice showed exaggerated liver injury. So these results strongly suggest liver CD25 positive ILC1 have a protective role in the CCL4 induced acute liver injury. So to determine the ILC1 or NK cells are important, our collaborator, Tim O'Sullivan in UCLA, performed the very important experiment. So he injected CCL4 into wild type or ZB683 deficient mice that severely reduced in liver ILC1 or NCL1 Curie EMS flox mice, which lack all NK cells, but still the ILC1 present in the liver. As shown here, reduced ILC1 mouse show worse liver injury after CCL4 injection. However, the no NK mice has no differences. These results clearly demonstrate liver ILC1s, but not NK cells, have a protective role in CCL4 induced acute liver injury. As I explained in the introduction section, ILC1 primarily produced interferon gamma after activation as a function. That's why we simply injected the CCL4 into interferon gamma deficient mice. As we expected, the interferon gamma deficient mice show exaggerated acute liver injury after CCL4 injection. So taken together with the prior experiment, interferon gamma has a protective effect, and this interferon gamma is secreted by liver ILC1 in CCL4 induced acute liver injury. So the answer of the very first question is yes. And then the activated liver ILC1 producing interferon gamma has a protective role in the acute liver injury. So question raised here is, what factors are critical for interferon gamma production by liver ILC1? So I'm an NK cell researcher, that's why I was very interested in the function of NK receptors on the cell surface of ILC1. As shown here, ILC1 expresses several kinds of activating NK receptors. That's why I focused the activating NK receptor DNAM1, CD226. And CD226 was originally identified the activating receptor which controls the cytotoxicity of NK cells. And after that, we reported the DNAM1 is important for interferon gamma production by NK cells and CD8 positive T cells. That's why we speculated the CD226 is also important for interferon gamma production by liver ILC1. Before testing the interferon gamma production by ILC1, we confirmed the expression of DNAM1 on liver ILC1. And very interestingly, liver ILC1 very highly expressed DNAM1, and the expression level is higher than NK cells. And then to test the function and the contribution of CD226 to interferon gamma production by ILC1, we injected the CCL4 into wild type and CD226 DNAM deficient mice and see the interferon gamma production by liver ILC1. As shown here, the DNAM deficient ILC1 cannot produce the interferon gamma efficiently as compared with wild type ILC1. Here is the quantified data. CD25 positive ILC1 are activated and predominantly produce the interferon gamma. We could see the CD25 positive ILC1 in DNAM deficient mice shown in blue bar, but the interferon gamma production is very inefficient as compared with wild type CD25 positive ILC1. That's why DNAM is critical for interferon gamma production by liver ILC1 after CCL4 injection. And then we confirmed that DNAM is highly expressed on ILC1. So how about the ligand expression? So DNAM ligand is CD155. This is highly expressed in the liver in the naive state. But after CCL4 injection, this CD155 DNAM ligand expression is strikingly upregulated in response to CCL4-induced liver injury. And then, eventually, we confirmed the ALT liver injury levels. As I explained, DNAM deficient ILC1 cannot produce interferon gamma. And then we know interferon gamma is has a protective role in acute liver injury. And then as we expected, DNAM1 deficient mice show worse ALT, worse acute liver injury. That's why DNAM1 has a protective effect, probably control the interferon gamma production by liver ILC1 in CCL4-induced acute liver injury. To directly confirm the DNAM1 activates and promotes the interferon gamma production, we isolated the NK cells and ILC1 in the liver and then stimulated with DNAM1 by using the specific antibody and cross-linking stimulation, as shown here. And see this and this. So when we primed the ILC1 with IL2 and IL7 and stimulated the DNAM1 by specific antibody, DNAM stimulation promotes interferon gamma production by liver ILC1. These results indicate that DNAM promotes interferon gamma production by liver ILC1. So we know the intrinsic factor DNAM is control the liver ILC1 derived interferon gamma production. But we also tested the extrinsic factor to control interferon gamma production by liver ILC1. Identification of dumps is often important to analyze the local immune responses, especially the tissue injuries. And then this time we used acute liver injury model. That's why we focused extracellular ATP as a dump, because liver is a very mitochondrial rich organ, and the 20% volume is mitochondria. And then it has a very high OX-phos activity. So that is the ATP synthesis is very high levels. That's why we speculated the extracellular ATP levels gonna be very high after liver injury. To test this, we collect the blood from the hepatic vein and then analyze and measure the extracellular ATP levels in the plasma. As shown here, the extracellular ATP level is strikingly elevated in the liver after CCL4 injection. So how about the receptor things? NK cells did not respond to CCL4 injection, but the ILC1 respond. So that's why we speculated the ATP receptor is exclusively expressed on ILC1, but not NK cells. And then we found the ATP receptor P2RX7 is exclusively expressed on liver ILC1, but not NK cells. And to investigate the role of P2RX7 and the ATP signaling axis, we used the BBG, P2RX7 selective antagonist. We injected this antagonist into the mice and then injected CCL4 and evaluated the interferon gamma production by liver ILC1. As compared with the vehicle controls, BBG treated mice, liver ILC1 in BBG treated mice could not produce interferon gamma efficiently. Here is a quantified data in blue bars. BBG treated liver ILC1 could not produce the interferon gamma efficiently as compared with red bar, vehicle control. That's why these data indicate that an ATP and P2RX7 axis is required for the optimal interferon gamma production by liver ILC1 after CCL4 injection. And then these mice did not produce interferon gamma. Consistent with our prior data, these mice show worse acute liver injury levels. To directly address the ATP effect on interferon gamma production, we isolate liver ILC1 and NK cells and stimulated with ATP. But very disappointingly, ATP alone did not stimulate the interferon gamma production by ILC1 at all. However, we combined ATP and IL-12. We could see nicer interferon gamma production by ILC1. So these results demonstrate that ATP alone is insufficient to promote interferon gamma production, but ATP accelerates IL-12-driven interferon gamma production by liver ILC1s. And then we also confirmed the IL-12 is produced by CD11B negative DC, which consists of CDC1 in the liver. As shown here, after CCL4 injection, IL-12 messenger RNA expression is highly upregulated in CD11B negative DC subset. Okay, so the answer of the question is activating signal through DNAM1 and then P2RX7 are required for the optimal interferon gamma production by liver ILC1. So question raised here is, how does interferon gamma ameliorate acute liver injury? So as you know, interferon gamma is a pleiotropic protein. And then through the literature, interferon gamma is reported to exaggerate or ameliorate acute liver injury. But 70 to 80% literatures report the exaggeration, and then only 10% or less show the interferon gamma is ameliorate the acute liver injury. We are not sure what's the critical factor, but the known mechanisms of interferon gamma on exaggeration or amelioration of acute liver injury is listed up here. So in our case, we are not sure which is correct. That's why we tested all of 4, but I could not see any differences in these 4 factors after the neutralization of interferon gamma by my hand. That's why we had to explore the new effect of interferon gamma on the liver. But the previous studies did not say anything about the direct effect of interferon gamma on hepatocytes. That's why we tested the hepatocytes. Very interestingly, when we analyzed the primary hepatocytes in CCL4 injected mice, we could see upregulation of Bcl-xL, which is a major survival factor, anti-apoptotic factor. However, when we injected CCL4 into interferon gamma deficient mice, we could not see any upregulation of Bcl-xL in the hepatocytes. So these results suggest the interferon gamma is required for the upregulation and maintenance of the expression of Bcl-xL in the hepatocyte after CCL4 injection. To directly test the hypothesis, we established the primary cell culture of the hepatocyte and then tested this interferon gamma, effect of the interferon gamma on Bcl-xL. In the big control of CCL4 DMSO, we could not see any impact on interferon gamma. When we compare the negative interferon gamma and positive interferon gamma in the culture, so there is no differences in the expression levels of Bcl-xL. However, when we culture the primary hepatocytes in the presence of CCL4, which is mimicking the in vivo situation, the Bcl-xL level is decreased probably as compared with DMSO. But when we culture the primary hepatocyte in the presence of interferon gamma, the Bcl-xL levels is maintained. It's not downregulated. So these results clearly demonstrate that interferon gamma helps the survival of damaged hepatocyte through the upregulation of Bcl-xL. We do not show the results due to time limitation. So Bcl2, another anti-apoptotic factor, is also show the same kinetics. So the answer of the question is interferon gamma contributes to the survival of damaged hepatocyte by upregulating Bcl-xL and probably also Bcl2. A remaining question is interferon gamma released from ILC1 sufficient to ameliorate the acute injury or not? To test the possibility, we tried to reconstitute liver ILC1. We saw that the liver ILC1 and then transplant the donor ILC1 with IL-15 via portal vein into RAG2 deficient, gamma2 deficient mice, which lacks all lymphocyte. As shown here, there is a donor, and the RAG2 and IL-2 receptor gamma deficient mice has no lymphocyte in the liver. After the transfer on 7 days, ILC1, donor ILC1 can completely reconstitute in the recipient mice. By using the mice, we injected CCL4 after the ILC1 reconstitution in the liver. And then, like viral type mice, donor ILC1 upregulated CD25 and CD25 positive ILC1 preferentially produced interferon gamma after CCL4 injection. And then now it's showtime. So we evaluate the liver injury. Without transfer of the ILC1, the recipient might show very severe acute liver injury. And then ILC1 transfer strikingly ameliorates the acute liver injury. But when we inject anti-interferon gamma neutralizing antibody in ILC1 reconstituted mice after CCL4 injection, this protective effect was abolished. That's why liver ILC1 derived interferon gamma is sufficient to ameliorate CCL4 induced acute liver injury. So fundamentally, this is everything in my study, but the summary is ILC1 derived interferon gamma has a protective role in acute liver injury. But many audience and reviewers ask us, this would be a very specific condition. So that's why they often ask us, can we generalize the finding into other acute liver injury models or something? And then we test that. The another acute liver injury models by administration of acetaminophen. Actually, in the US, the acetaminophen is one of the greatest cause of drug-induced acute liver injury in ER. And then the pathogenesis is almost same as CCL4. So this is drug-induced acute liver injury model. And then we tested low-dose acute liver injury models by RAG1 deficient mice. And then similar to CCL4 induced acute liver injury models, ILC1, but not NK cells, upregulated CD25 in response to the AP injection. And then CD25 positive activated liver ILC1 preferentially produced interferon gamma. And then when we deplete these activated CD25 positive ILC1 by using the CD25 depletion antibody, these depleted mice show worse acute liver injury. That's why in another acute liver injury models, liver ILC1 have a protective role. So here are the conclusions. ILC1s have a protective role in the acute liver injuries. And then activated liver ILC1 produce interferon gamma. And then at least in CCL4 induced acute liver injury models, so DNAM1 is required for the optimal interferon gamma production, and the ATP as a dump accelerates IL-12 driven interferon gamma production. And then mechanistically, interferon gamma helps the survival or the damaged hepatocyte through upregulation of Bcl-xL and Bcl-2 probably. Here is the graphic abstract, and we successfully reported the results in the Immunology 2 years ago already. And then this is the last slide. So I greatly appreciate the helps by many of our collaborators, especially team in UCLA. And then this study was supported, financially supported by many foundations. And thank you for your introduction. And then I'll be very happy to take any questions.
Hello, Nabekura. Really great talk. And I really enjoyed hearing about the story.
Yeah, thank you.
Obviously, have lots of questions, but I'm going to start with the kind of bigger picture questions. So I think you did a really nice job introducing some of the history of how ILCs were discovered and the different subsets of ILCs and what roles they play. And you're looking at acute injury. I had a question about maybe post-acute injury, maybe in regards to resolution or tissue.
Oh, yeah.
When you're repairing the tissue after acute damage.
Mm-hmm.
You think other ILCs are also playing a role? For example, ILC2s are now coming later in your CCL4 model, for example, and maybe helping resolve the tissue injury.
Yeah, so probably the ILC2 would be the critical to induce the chronic acute liver injury. Especially they promote the fibrosis. And then historically, interferon gamma is an anti-fibrotic cytokine. That's why in the chronic situation, probably ILC1 and also circulating and recruited NK cells also produce interferon gamma and contract to the ILC2 derived IL-4 or IL-5, IL-13 fibrotic type 2 immune responses. But in terms of this contract action of interferon gamma by IL-1 and NK cells are not reported yet. Yeah, but probably that's true. And then by our hand, we didn't test it yet.
Okay, I think Philip has a question, and I saw some other questions, or Philip has some questions in the YouTube chat. Do you mind discussing those, Philip?
Yeah, sure.
Sorry,
so the first question was just regarding the source of ATP. So in the intestine, the microbiome has been suggested to be a source. So does this model, do you get pathology in a germ-free mouse? Or could there be a role for the microbiome, even if not making the ATP, but actually being a trigger that leads to cell death or something that leads to release of ATP?
Well, honestly saying, I'm not sure. And then we did not test the ATP levels in germ-free mice or antibiotics-treated mice. But what we did is the measurement of the hepatic bay plasma ATP levels. And then when we analyze the portal vein ATP levels, probably the intestine or microbiota may have an impact. But yeah, as I said, we are not sure. And then as compared with the naive levels, so after the CCL4 injection, so the plasma ATP level is super high, micromolar level. That's why, so the microbiota may regulate the basal levels in naive state. But yeah, I'm actually not sure the microbiota can control the levels of after CCL4 or acute liver injury. Yeah, that's my answer.
The other question I had then was with regard to the protective effect of interferon gamma. So do you think that they have a particular, the ILC1s have a particular localization, or is the tissue itself just primed to respond to interferon gamma in a way that's beneficial for the tissue?
So in terms of localization issue, we also tried to identify the location or spatial temporal position of the ILC1. But the technical issue was we don't have any ILC1 specific marker. So and then NKp46 is the nice marker for ILC1 and NK cells, but we have to merge with the ILC1 specific marker or NK cell marker. But that's very hard to find. And then probably Tim also tested. And then I asked tried to collaborate with the histology guy in UCSF, but he actually failed to find the ILC1 but not NK cells in the liver by histological section. Yeah, that's the issue.
Yeah, it seems like a kind of a pretty challenging problem, but it is interesting to think that, they may localize to a certain place, and, how that would happen, I don't know, but that they would deliver.
Yes.
This was great. Thank you.
I had one more question. In regards to DAMs, in addition to ATP, do you think other ones are playing a role? For example, HNGB1 or other?
Oh yeah. So in this experiment, so we actually add HMGB1, IL-33, IL-1 alpha, and hyaluronic acid or something in vitro in the culture of ILC1. But even if we combine with these dumps with IL-12, there is no differences in IL-12 plus combined with these dumps with IL-12. Only ATP can accelerate the IL-12 driven interferon gamma. That's why from the result of this experiment, so we decided ATP has the greatest impact as a dump after CCL4 injection.
Well, that's really cool. Thank you. Okay, I don't see any other questions in the YouTube comments. So I'm not sure if Philip has any last questions.
No, I'm good.
Yes, I think we'll conclude your seminar, but I just want to say thank you very much for providing this really awesome talk about your work, and I look forward to future talks from you and seeing future updates on this current work. It's really great. So just want to thank you and also thank all of our attendees and viewers. We look forward to a new round of talks in 2023, and I wish everyone the best and happy holidays.